Chapter 5, Membrane Structure and Function. As we recall, the plasma membrane of all cells is primarily composed of phospholipids. Phospholipids are the perfect molecule for composing the plasma membrane because of their status as amphipathic molecules. They have a dual nature. On one end, they have these phosphate heads, and on the other end, they have two fatty acid tails. The phosphate heads are hydrophilic, water-loving, and so we can see that on the external surface of the cell, we have all of these hydrophilic polar heads facing the aqueous environment outside the cell, and all cells, whether they're part of a living organism or whether they're free living in the environment like an amoeba, face an environment that is mostly water. In other words, they live in an environment that's mostly water. And on the other side of the plasma membrane, we have another layer of phospholipids with the heads facing towards the cytoplasm inside the cell. The cytoplasm, as you recall, refers to both the liquid called cytosol that makes up the interior of the cell that everything floats in, you might think, or you might say, and therefore it's perfect that these hydrophilic heads face the inside of the cell. So as we've reviewed in class previously, if we point to, as I'm doing with the pointer now, the internal portion of the plasma membrane between the external and internal layers of hydrophilic polar heads, the phosphate heads, we have a hydrophobic region. And so it's essential that we remember that this region, as labeled here, is hydrophobic, water-hating, whereas outside the cell and inside the cell are both water-loving regions because they're primarily composed of water molecules. Now, when we talk about the structure of the plasma membrane, we typically use the term fluid mosaic model. And so fluid refers to the fact that all of these parts of the plasma membrane have some flexibility to change and alter their location within the membrane. Now, that's not 100% true for all of these. Some things are anchored in place. And so as we look at these diagrams from our text in the bottom portion of this slide, what we notice is we see a number of components that are embedded or attached, in some cases embedded and in some cases attached to this plasma membrane. Most of these are proteins. Some of them have sugars attached, making them glycoproteins, but we typically use the following terminology. We describe the proteins as icebergs floating around in this plasma membrane. And so the proteins float in the plasma membrane like icebergs float on a sea. So they move around slowly from place to place, and in some cases they are constrained by being attached to other proteins that restrict their movements. What we notice is this, that proteins may be peripheral or integral. And so integral proteins are proteins that span the plasma membrane. We can look over here at this diagram on the right, and we see an integral protein. And so it goes from one side of the membrane to another. Oftentimes, an integral protein will be a channel that will function, in other words, as a channel, and so it allows something to move from one side of the membrane to another. That doesn't have to be the case, but that may be the case. Peripheral proteins, on the other hand, which are shown in this diagram on the right and in this diagram on the left, are proteins that are located on the edge of the plasma membrane. They're not embedded in the membrane and therefore are not intimately part of the membrane. In some cases, what we notice and see in this diagram is that we have carbohydrate chains that are attached to these proteins, making glycoproteins. In some cases, these carbohydrate chains serve as a sort of name tag that's recognized by the immune system, allowing the immune system to distinguish self from non-self, which is a big deal in biology, as you would imagine. Your immune system is constantly roaming throughout your body looking for cells that do not have the correct name tag. When they find those cells, they will attack and hopefully destroy those cells. And so that's basically how your immune system works. You're aware of the fact that sometimes diseases called autoimmune disorders can occur. In the case of an autoimmune disorder, the immune system becomes confused and it forgets that it should recognize one of these name tags, and it sees the name tag as foreign that it should not. So it sees part of your sees a normal cell in your body as foreign and attacks that cell. Lupus or rheumatoid arthritis would be good examples of autoimmune disorders. Lastly, what we see is that cholesterol is a frequent component of the plasma membrane. And so we see it here in the diagram to the right, and also the one to the left drawn in purple. Cholesterol impacts the fluidity of the membrane, allowing the membrane to remain flexible in all conditions. One thing that we notice is that organisms referred to as psychrophiles, organisms that reside in very cold environments, including right directly on ice and snow, have a higher concentration of cholesterol in the membrane, therefore allowing the membrane to remain fluid or flexible at those very cold temperatures. Now, we move on here to talk about proteins that are found in the membrane. And proteins that are found in the membrane do some exciting things. And we have 6 examples that are found on the screen. So we'll talk briefly about some examples of each of these, beginning with A. So in A, we have a carrier protein. And we have a channel protein. Starting in A, we have the channel protein. And in the case of a channel protein, we're allowing a particular molecule or ion to cross freely across the membrane. Now, most notably in the category of channel proteins are ion channel proteins. And so ion channels play a tremendous role in the nervous system, as you're likely aware. In fact, any sensation that you experience, the fact that you can hear me right now, the fact that you have thoughts and emotions for that matter, is all a result of these channel proteins and the movement of the ions, sodium and potassium, back and forth across the neurons of your body, which is a fascinating topic and one that we'll talk about more in the future. Moving on to B, the second type of membrane protein. We have carrier proteins. And so carrier proteins are proteins that selectively bind to a single molecule and move that molecule from one side of the membrane to another. And so you may ask, well, what's the difference between a channel protein and a carrier protein? And the difference is this. It's often described as the difference between a bridge and a ferry. And so all of us know what a bridge is, and hopefully you know what a ferry is as well, F-E-R-R-Y, ferry. And so a ferry is a boat that stays at one side of the river, and a car pulls up, drives onto the boat drives across the river and drops the car off on the other side. And so that's analogous to a carrier protein. In other words, a carrier protein picks up, as we see in this diagram, one particular molecule from one side of the membrane, and it drops it off on the other. And so it has to undergo a conformational or a shape change. Another way of putting it would be a shape change as it moves that molecule or that ion possibly from one side of the molecule to the other. Whoops. And so carrier proteins are much, much slower than our channel proteins. It should be noted that both channel proteins and carrier proteins are specific, so they're probably letting just one type of molecule or one type of ion cross, but a channel protein is like a bridge. It opens and cars go back and forth. In other words, it opens and ions go back and forth very, very rapidly, huge numbers in short periods of time. Carrier proteins, much, much slower, like the ferry. The car pulls up, takes it across, and gets the ferry takes it across, and it gets dropped off on the other side. So those are carrier proteins. Next, we have the cell recognition proteins, and so these are the glycoproteins, the carbohydrates that we referred to just a moment ago. And so the most prominent example of these are the so-called MHC glycoproteins, the major histocompatibility complex glycoproteins. These are essential in allowing the body to recognize and distinguish between self and non-self. And so there are many interesting aspects of this, including the idea that certain parts of your body, such as your inner eye, are excluded from these surveys that the immune system does early in development. And so should your immune system ever get access to the inner part of your eye, it will attack it because it recognizes that part of your body as foreign. And so cell recognition proteins are very important in allowing the body to determine which cells are supposed to be there and which cells are not supposed to be there. We move on to D, receptor proteins. And so receptor proteins will be familiar to you because of our discussions of hormones. Remember that a couple class periods ago? So receptor proteins are don't exclusively play a role in hormones, but they also are involved in neurotransmitters, for instance, in the body, and some local signaling factors that don't travel throughout the body like hormones do, but do travel from one cell to another. And so in the case of a receptor protein, you have a protein that's integral in this case, and it has a special shape there that's complementary to another molecule referred to as a ligand that binds to it. And so you can see the ligand binding to the receptor protein, and when that occurs, something happens, which is not shown in this diagram, but down on this side, if you look at the pointer, something is happening, and so something takes place in the cell as a result of that binding. Hormones always work in this manner, with some small exceptions. There are hormone receptors that are sometimes found in the nucleus and the cytosol, but many hormone receptors, especially hormones that are hydrophobic, like the steroid hormones, they cannot cross the plasma membrane. Their receptors must be found on the exterior of the plasma membrane. Next, we have enzymes, and so excitingly, sometimes enzymes are found embedded in the plasma membrane. We'll certainly have a whole chapter devoted to enzymes soon, but for now, it's sufficient to say that we do find some enzymes that are located in the membrane, and of course, enzymes bind to a substrate, turning that substrate into a product. The full discussion of that goes a little bit beyond this particular chapter, but we do return to it in, I believe it's chapter 6, if I'm not mistaken. Enzymes are certainly found in other places as well. In fact, the minority of enzymes, would be found in the plasma membrane, but there are enzymatic proteins associated with the plasma membrane. And then lastly, but not least important, we have junction proteins. And so junction proteins are responsible for binding one part of one plasma membrane, shown here, to a separate cell, the plasma membrane of a separate cell found here. Tight junctions are a good example of junctions that bind cells tightly together. In fact, not allowing anything to pass between the cells. You find tight junctions, for instance, in your skin, which makes your skin quite waterproof overall. And so there are other types of junctions as well, but it is quite possible that proteins associated with the membrane do function as junction proteins, binding one cell to another. Here we find a summary of all those types of proteins that we just talked about, and of course you're going to be reading about those in your text as well. And here we enter into a short discussion of the permeability of the plasma membrane. And this is an important idea. As we established in lecture on Wednesday, we realized that the plasma membrane is selectively permeable, or sometimes we refer to it as semi-permeable. In other words, it doesn't let everything through, but it does let some things through the plasma membrane. So what we notice is there are two things to consider if you're going to, as to whether or not you can get through the plasma membrane. One thing is size, and one thing is charge. Size and charge. So the way it works is the following. Here we have a small non-charged molecule. So in other words, this is a nonpolar molecule, meaning it is not soluble in water. It's a hydrophobic, a water-hating molecule. That means it can slip easily into the interior of this plasma membrane, and it can move from one side of the membrane to the other with no problem whatsoever. It's a small molecule, and it has no charge, and so it has no problem moving across the membrane. On the other hand, we can look at a large molecule. So here we have a protein, and so it, generically labeled macromolecule. But proteins, as we mentioned, are always big. We're talking about, certainly greater than 100 amino acids in length. And so it's likely that the charge is going to prevent it from getting through the membrane anyway, but big molecules, large macromolecules, have a hard time making their way through the membrane. So they're typically impermeable to the membrane. Now, on the other hand, over here, we have ions. And so these ions, of course, are charged. By definition, an ion, of course, is a charged atom. And what we notice is that this charge prevents it from making its way into this non-charged region, this hydrophobic, water-hating region of the plasma membrane. So even though the ions are tiny, they make their way to the plasma membrane and they are rejected. And of course, all of our physiology depends on this. If ions could move across your membranes, you would cease to exist instantly. Because every single nerve cell in your body, of the 80 billion that make up your brain, for instance, relies on this principle of selectively allowing ions, primarily sodium and potassium, to move across this membrane at the appropriate time. And so, of course, they need to not be allowed to move across any old time they want. Now, lastly, we have one that's a little bit perplexing, water. So as you know, water certainly is a polar molecule. However, what we notice is that water can move across the membrane. And so it's tempting to say, well, okay, water's a polar molecule and it's pretty small, so that lets it go across the membrane. And that's not entirely incorrect, but it's useful to note that water is permeable to the plasma membrane primarily as the result of these proteins referred to as aquaporins. Aquaporins are little channels for water that exist in the plasma membrane of all cells. Some cells, like the cells of your kidneys, have the ability to modify the number of aquaporins that are present in the plasma membrane, therefore adjusting the overall permeability of the plasma membrane to water molecules. For instance, you can adjust to a large extent the concentration of your urine, and that's done largely by the insertion of aquaporins into the kidney cells in the distal tubule, and therefore that allows you to adjust how much water is reabsorbed and how concentrated the urine is going to be. Now, we leave this picture and this topic to go over here to passive and active transport. So what we notice is this. In the case of passive transport, no ATP is required and molecules follow a concentration gradient. By this, it's essential we understand that we mean that molecules are going down a concentration gradient. They're moving from an area of high concentration to low concentration, which they do without any additional energy input as they randomly move and spread out from a concentrated to a non-concentrated area. Active transport, in contrast, needs two things. It requires ATP, which is not listed here, and it requires a carrier protein. And so we just talked previously about carrier proteins. And so in the case of active transport, it's like pumping water back up above a dam. You grab a molecule and you take it from an area of low concentration, you take it to an area of high concentration. So passive transport, no ATP required, molecules go downhill as far as concentration gradient goes. Active transport, ATP is required, molecules go uphill. And it does require energy. Sorry, that is on there. Guys, this is where we picked up on Wednesday with our discussion of osmosis, and so what we're going to do is flip right through these slides that we've already covered, all these things we covered on Wednesday. So you're familiar with these terms, you did well with these in lab, and we find ourselves now at some types of transport that require carrier proteins. So it's appropriate that we jump to this particular topic at this time because what we see is that carrier proteins can be used with energy or without energy. Now, facilitated transport is this. It's a special type of diffusion. Facilitated transport occurs when you have a molecule that wants to go down a concentration gradient, in other words, from an area of high concentration to low concentration, but for one reason or another, and those two reasons will be size and charge, they cannot make their way through the membrane on their own. Therefore, we have a carrier protein that binds to them on one side and dumps them off on the other side, allowing them to move down their concentration gradient. But still, it's important to note, in facilitated transport, no energy is required. It's sometimes called facilitated diffusion. It is a form of diffusion that requires a transport protein. In active transport, as we just discussed, molecules are moving against a concentration gradient or up a concentration gradient from an area of low concentration to an area of high concentration. Molecules must combine with carrier proteins. There's no other way to do it, and in this case it does require energy because the molecules will not move in this direction on their own. So here we have, if we were in class, I'd ask you what's being illustrated here, and you would say facilitated transport or facilitated diffusion. High concentration of molecules here, carrier protein, the molecule binds to the carrier protein changes shape, the carrier protein dumps the molecule off on the other side, and everyone's happy. So we're going from an area of high to low concentration, therefore we do not need energy, no energy required. Again, this is an example from your book of facilitated diffusion. Now we have an example of active transport. In the case of active transport, as you know, we're pumping things from an area of low concentration to high concentration, and so it does require energy. One of the best examples, well, maybe I should not say that, one of the most prominent examples in the body of active transport is the sodium-potassium pump. So in the case of the sodium-potassium pump, we have a carrier protein, and the carrier protein has 3 binding sites for sodium on this side and 2 binding sites for potassium on this side. And so what we notice is that the sodium goes into the cell, into the carrier protein, and it binds to the binding sites that are found here, little squares. Now let's advance one slide. So now we're at number 2 here, and what we see is that ATP is expended at this particular point. ATP, as you know, the energy is stored in the phosphates. We cleave that third phosphate, transferring it to this so-called sodium-potassium pump. We're left with ADP here, which doesn't have a lot of energy. The result of that expenditure of energy is a change in conformation of the carrier protein. The 1, 2, 3 sodiums are pumped outside the cell, and at this point, no additional energy is required. 2 potassiums enter the sodium-potassium pump, binding to the binding site there, and then it rocks back to its original position, losing this phosphate as it goes, dropping off the potassiums, as you can see here, inside the cell, and the whole thing happens again. And so this particular sodium-potassium pump is found in every single cell of your body, and in every single cell of your body, it completes the cycle that we see on the screen. It moves 3 sodium outside for every 2 potassium it moves inside. Both of these ions have a charge of +1, so the result is that you have a greater positive charge outside than inside, and this is one of the factors that contributes to the negative charge that we find in every single cell of the body, most notably, and perhaps most importantly, in the nerve cells of the body, and then right behind that, the muscle cells of the body. In fact, in the nerve cells of your body, you apparently maintain sodium concentrations that are somewhere in the neighborhood, that are thousands of times greater, somewhere in the neighborhood of thousands of times greater outside the cell versus inside. And as we mentioned previously, any sensation that you experience, pain, not a pleasant one, but one that we're familiar with, is the result of the movement of sodium across the cell, not via this manner, but in other words, the sodium is stacked up outside the cell, and then there's sodium channels that allow it to go back in, but it wouldn't go back in if it wasn't pumped out via this sodium-potassium pump and via active transport. So everything about your neurophysiology and muscular physiology too relies ultimately on this sodium-potassium pump that is active in your body right now in every single cell. Now, lastly, we want to ask ourselves the question, how do we get large molecules back and forth across the cell? So what we just talked about is good for ions or very small molecules, like glucose would be fine, but how do we get carbohydrates, big carbohydrates back and forth across the cell, or big bunches of molecules, or parts of the cytoskeleton, or even things like large protein hormones, like growth hormone? How do we get those back and forth across the cell? And the answer is that we use 2 processes, exocytosis and endocytosis. Exocytosis, which moves items out of the cell, endocytosis, which moves items into the cell. In the case of exocytosis, what we notice is that vesicles fuse with the plasma membrane and they dump their contents outside the cell. You've all seen soap bubbles, and you're aware of the fact that when 2 soap bubbles run together, they merge, forming a larger bubble. So as you envision exocytosis and endocytosis, that's what you should be thinking. Imagine a vesicle, which is simply a ball made of plasma membrane. The vesicle contains something. So let's say it contains growth hormone, which is released in this manner. Growth hormone is found, the growth hormone containing vesicle is currently found in the cytoplasm. It runs into the plasma membrane, and as it runs into the membrane, it fuses with the membrane. So now, all this growth hormone, which was inside the vesicle, finds itself outside the cell, and it's exactly how growth hormone is secreted, via exocytosis. Not only that, but even crazier still, as you go from one neuron to another neuron, or from one neuron to a muscle, then you have neurotransmitters that are released in this manner, allowing a nerve impulse to move from one nerve to another, or a nerve impulse to move from a nerve to a muscle. So there's not a movement that you made during the course of your life, a muscular movement, that was not the result of this exocytosis. Endocytosis is just the opposite. And so in the case of endocytosis, you have this invagination of the cell wall that occurs. It gets larger and larger, and then that breaks off, forming a vesicle that moves into the cell. There are 2 types, though, in this case. The first is phagocytosis. And so in phagocytosis, what we notice is that a particular large item or items are taken in via endocytosis. Phagocytosis is often specific because the cell is pursuing a particular item or responding to a particular item it wants to engulf outside the cell. And it's also worth noting that all cells do not carry out phagocytosis, only some cells. And so we do notice that amoeba carry out phagocytosis. We notice that many of our white blood cells, or at least several of our white blood cells, would carry out phagocytosis. But not all cells certainly carry out phagocytosis. So it's worth noting that some of your white blood cells, when you have an infection, they pursue the bacteria that are causing the infection, engulfing the bacteria, and then they engulf it via phagocytosis, and then they die in most cases, and more white blood cells are formed. Pinocytosis is a little bit different. Pinocytosis is often equated or referred to as cell drinking. So in the case of pinocytosis, the cell forms very small vesicles that break off, and a little bit of the plasma membrane makes its way into the cell. It's important to note that all cells carry out pinocytosis. Not all cells carry out phagocytosis, but all cells carry out pinocytosis. What does the cell get out of pinocytosis? Well, it gets a little bit of whatever it is that's in the solution around it, and cells benefit from gaining nutrients and sometimes other molecules that are needed in this particular fashion. Lastly, we have receptor-mediated endocytosis. So this is different. It's kind of similar to pinocytosis, but it occurs when a receptor binds, or excuse me, a ligand binds to a particular receptor that's found in this so-called pit located on the plasma membrane. When the ligand binds to the receptor, it triggers the formation of a vesicle, and what the cell wanted was that ligand that was binding to the vesicle. So upon binding, a vesicle forms. Let me restate that. In the case of receptor-mediated endocytosis, we have a ligand that binds to a receptor on the plasma membrane, and a vesicle forms in response to that. And so what the cell originally wanted was that ligand that's binding to the receptor. So let's go to our next slide, and we'll see some examples. And so here we have exocytosis, and so this could quite likely be growth hormone, for instance, in the little proteins, little green spheres that are found inside the vesicle. It makes its way to the plasma membrane, merges with the plasma membrane, and dumps the contents outside the plasma membrane. Exocytosis is incredibly common in the body. Next, we have endocytosis. And so endocytosis, as we mentioned, occurs in three ways. Here's phagocytosis, and we see one large particle being engulfed. Here we see an amoeba that's on its way to forming a food vacuole as it pursues this little ciliate that it's going to eat. Here we have pinocytosis. As we said, all cells carry out pinocytosis. Here's an electron micrograph of a cell forming these vesicles as it carries out pinocytosis. Lastly, we have receptor-mediated endocytosis. And so we have these receptors found here. Here's the ligand, labeled as solute there. It binds to the receptors and triggers the formation of this vesicle. And as the vesicle forms, it takes the little molecules that the cell was interested in the first place. The last topic that we have are cell junctions. And so, interestingly, what we see in this particular case is that we have three types of junctions to which we want to address as we close this particular chapter. And so, the first type of junction that we have are adhesion junctions. And so, in the case of adhesion junctions, we look down here to the left-hand side of the screen, and we see that in an adhesion junction, we find a scenario in which we have this so-called plaque that's located, which is basically this big plate located inside the plasma membrane, and we have it located inside the plasma membrane of cell A, inside the plasma membrane of cell B, and then we have these little intracellular filaments that reach out and they connect these two plaques, therefore connecting the two cells right there in the middle. This is found in portions of the body in which we have a good deal of flexibility. And so we find this in portions of our internal membranes in which the membrane needs to stretch and move a great deal. And so there is some, we do allow some passage. This junction does allow some passage of materials between those two cells. The next type of junction we have are tight junctions. And so in tight junctions, we see that we have proteins that form these barriers that hold cells together extremely tightly. And so tight junctions exclude the movement of any molecules between them. And so we find tight junctions, for instance, in the lining of the stomach and in skin to some extent. And so the lining of the stomach, of course, must be very impermeable in order to ensure that the contents, the low pH of the stomach, doesn't make its way outside the stomach. And so tight junctions accomplish that feature very nicely. Lastly, and most excitingly, we have gap junctions. And so in the case of gap junctions, we have 6 little proteins referred to as connexins that are found on one side. We have 6 little proteins called connexins found on the other side, and they meet there in the middle, forming a channel that connects the cytoplasm of this cell, cell A, with the cytoplasm of this cell, cell B. And so ions can flow freely back and forth from one cell to another. Now we find gap junctions primarily in heart cells. In cardiac cells, what we see is that all of the cells on the top of the heart, in the atria, and on the bottom of the heart, in the ventricles, are connected to one another via these gap junctions. We also find gap junctions in some of the tissue in the small intestine as well. Gap junctions allow cells to work together in a coordinated fashion, as is necessary, of course, in the heart and in at least small patches of our small intestine.